Hello, for this presentation, I'd like to go ahead and give a quick little overview to one of my favorite bacteria, Staphylococcus epidermidis. So, Staphylococcus epidermidis is a gram-positive, coagulase-negative opportunistic pathogen. Um, the term opportunistic pathogen is similar to other species of staph, such as Staph aureus. Staphylococcus epidermidis is significantly less virulent, as it only has about 2 virulence factors, as opposed to Staphylococcus aureus, which we know expresses a wide array of different virulence factors. So it is a natural member of our skin microbiota and our skin flora. Here in my little representation of our skin cells, you can see Staphylococcus epidermidis living happily on the skin. It's very important for our skin flora because it can outcompete other much more infectious virulent bacterial cells such as Staph aureus. However, Staphylococcus epidermidis is an opportunistic pathogen and can quickly cause infection. For example, if we imagine a little cut into our skin right here, the cells can invade our epithelial skin layers and go to the bloodstream, causing sepsis and bacteremia. One of the main ways that Staphylococcus epidermidis causes infection is actually nosocomial infections or hospital-borne infections. So this image I have over here to the right is let's picture it as a zoomed-in needle or catheter, IV, central line, whatever you prefer. So what can happen is if not properly sterilized or you don't properly wash your hands, the Staphylococcus species can form on the needle, and Staphylococcus epidermidis has this ability to form these special layers called biofilms, which we'll discuss more in a second. And when those biofilms are introduced directly into the bloodstream, they can very, very quickly cause sepsis. And unfortunately, Staphylococcus epidermidis, like many other species of staph, for example, aureus or MRSA, it is naturally resistant to many different types of antibiotics, particularly ampicillins, penicillins. It is still a little susceptible to vancomycin, but every year they're finding more and more strains of the species that are unfortunately no longer susceptible. So to take a little more detailed look into the biofilms. So the two main virulence factors of Staphylococcus epidermidis are shielding and adhesion factors. So the adhesion is mainly done through proteins, which are expressed by the AGR gene. So proteins, and then the shielding is mainly done through polysaccharides. One of the main examples is polygamma glutamate acid, or PGA. So I'll write that out here. So what these do is they go ahead and they're mainly expressed when Staphylococcus epidermidis forms in large concentrations. For example, if we imagine this image down here as a zoomed in catheter, we can imagine all the little bacterial cells gathering and growing together and beginning to form a biofilm. So as this biofilm grows, what will happen is the AGR gene will be expressed on the outer layers, which this gene is what causes the adhesion and shielding factors to be expressed, which forms a capsule-like structure. So this biofilm, like everything included under the green, is extremely, it's almost capsule-like, and it is extremely resistant to our body's natural immune system, and it won't really recognize the biofilm, which is one of the reasons why biofilms are so dangerous. And the proteins and polysaccharides are very large molecules, and they are very hard to remove through just like standard cleaning. So a lot of times they'll go ahead and they'll sneak onto those medical devices. But once the biofilm grows large enough, what'll go ahead and happen is the biofilm will rupture, and then all of these bacterial cells are very, very quickly released directly into our body's bloodstream, which is very dangerous and can cause sepsis. And as we discussed before, unfortunately, antibiotics are not always helpful in these sorts of situations. So the main way we can help to prevent these infections is making sure proper sterilization of medical equipment, no reusal of medical equipment, no reusal of needles, and then making sure doctors and nurses are extra careful to wash their hands and wear gloves and overall use antiseptic measures. So that was just my brief introduction to Staphylococcus epidermidis. Thank you for listening.